I'll put out YouTube and Facebook. Don't worry about it. I'll post this.
You should have asked them.
Okay, quiet down, everybody.
You know, really can leave a sign of it. If you went online, it says you can post your pictures in videos anywhere. Either you could accept. And also anything you can do.
Okay, I'd like to present Dr. Uri Samuni from Queens College.
Can you hear me when I talk up there? Yeah. We need to speak. Can you hear me up there?
Yeah.
Okay. I just asked if you know this building. Some of the data here is not public here. Okay. So I'm going to tell you a little bit of what we do in Queens College, which is part of CUNY. If you're familiar with CUNY, it's the largest, I think probably definitely largest in the United States, or maybe even the largest in the world, public university system. We have 5 major or senior colleges. One of them is Queens College, which is in Flushing. And I'm going to tell you a little bit of one of the topics, one of the research projects that maybe 2 different research projects that we run in my lab. So what we do is interdisciplinary, but primarily it's biophysics. Okay? And the title of my talk is Soul Gel Matrices, a Biophysicist Sandbox. And I'll explain later why the sandbox. So we're interested in a variety of different topics. One of them is the structure-function relationship between proteins, which is how the structure brings about the function. We are interested in using different kinds of spectroscopy. One of them is Raman spectroscopy, and also resonance Raman spectroscopy, which is a very powerful kind of spectroscopy because it gives you structural information. And we're also interested in utilizing a method called soul gel encapsulation of proteins or biomolecules within matrices, which I will explain later. We also have a project where we're fabricating nanoparticles, and we hopefully will get to that by the end of the talk. And we have a separate project, or separate topic in the lab, a bunch of projects that involve free radicals and antioxidants, and some of our research now ties this project with the other project, but we're not going to be able to talk about it today. Next time. We'll keep something for next time. Okay. So, one of the things we study is proteins, and why do we want to study proteins? So, as you may know, almost everything in our body, in biological systems, involves proteins. Either as a structural, proteins are used structurally to build different structures in cells and so forth, or as an enzyme to speed up reactions. So if we didn't have proteins, reaction would take, instead of milliseconds, it'll take thousands of years, and life wouldn't proceed the way we know it. Okay. Now, if there's a problem with the protein, maybe a genetic problem, okay, or from other reasons, or poisoning and so forth, and the protein doesn't work properly, it doesn't do its function, then you have a manifestation of that is a disease. And our objective is to help people who have disease. So if we want to understand how we can approach a disease and improve a person's life, or maybe even cure it, we need to understand how proteins function. Okay. So in order to understand that, we need to know how the structure brings about the function. What changes in the structure are critical for the function. Okay. This is also of interest because a lot of companies and even universities are interested in different applications where you can utilize, you can harness the power of, say, proteins of their enzymatic ability. ability to catalyze reactions, you can harness that for all kinds of applications that people want to improve on. If in a chemical reaction you want to speed up the reaction, and an enzyme already does that, can you then utilize the enzyme for that? Okay? If you want to use an enzyme as a biosensor to detect something, for example, if there's a CO, or for example, sometimes people want to know if you sent a package, if it was ever exposed to oxygen. If it was sealed and something you have to make sure it doesn't get exposed to oxygen, okay, because then it goes bad. You can have a protein there that changes color when it's exposed to oxygen. Okay, so you can use that. So if you know if you properly understand the relationship to the structure and the function of a protein, you can then utilize it for all kinds of applications. And people are doing it. There are many applications already. So one of the systems that we studied is a protein called hemoglobin, which you may have heard of. All of you have it. Okay, you carry it with you. It's in your blood. That's the protein that gives the blood the red color. Okay. What it does, it's responsible for binding oxygen and delivering it to the tissue, and then taking CO2 and delivering it back to the blood. Okay. So hemoglobin is composed of four subunits. 1, 2, 3, 4. Each one of them is a polypeptide. So hemoglobin as a whole is a polypeptide. It's made of 4 peptides. Each peptide is a form that's globular form. It wraps around itself. And the center of each one of them is a heme group. This is the heme group 1, 2, 3, 4. In the center of the heme group, which is like a little disc, there's an iron. And that iron binds the oxygen. Okay? So in the red blood cells, we have billions of those proteins, and they bind oxygen reversibly, deliver it to the tissue. Each hemoglobin can bind a total of 4 oxygens, because there's 4 binding sites. 1, 2, 3, 4. This is now a cartoon that just shows you what the heme group looks like. Okay, so this is the heme. It's a flat vein. In the center of it, there's an iron. It's anchored to the protein using an amino acid called histidine. So here, the histidine is bound to the iron. And that's what holds this whole unit inside the protein. Okay? Now, this is when you have no ligand, there's no oxygen. So we call it a deoxy form. Deoxy, there's no oxygen. Okay? This is the hemoglobin when it's in your lungs, before it's acquired with any oxygen is bound to it. And oxygen arrives and binds to the iron. Binds to the iron, and it pulls the iron a little bit up. Now, in this deoxy form, the iron is outside, a little bit outside of the plane of the heme. So this is the heme, the iron is sticking out a little bit. When the oxygen comes from here, it pulls it up. It pulls it up, and it pulls the histidine with it. Okay, it moves it maybe, I think about 0.2 nanometer. That's it. That's the movement. And this little movement brings about a big change. It then moves. The oxygen comes and moves the iron over here. The iron then pulls the histidine. The histidine moves this whole helix. And this whole helix causes the whole protein, which is hundreds and hundreds of atoms, to rotate like this. Okay? So this is known. Okay? And we know that the structure of the protein is known. It's known from X-ray structure. And we know that there's one structure when it's not bound to anything, and we call it the T structure. And there's another structure that's called the R structure, where it's bound. And we know that it goes from this structure to this structure when it binds oxygen. And elucidating this structure is one of the first X-ray structures that was ever acquired. And people who did that won the Nobel Prize for that. And the people who came up with the model that explained this, the T and the R structure, won another Nobel Prize for that. Okay? And all of protein, almost everything that we know about protein is based on these initial studies. Okay? But even though this was done right, say probably in the mid-50s, okay? To this day, there's not even one full description of what happens when you go from here to here. Okay? What we do is we study it spectroscopically. We use spectroscopy, and this is resonance Raman, and we have a spectra here of the hemoglobin when it's not bound to iron, to oxygen, and here when it's bound to a ligand, in this case it's CO. And there's a peak over here, which is the iron histidine peak, which has a frequency of 214 when it's not bound, and 230 when it's bound. So we have a spectroscopic marker that we can look at it and we can interrogate our sample and know if it's bound or not bound. Okay? And we know that this is what you see when it's not bound, and this is what you see when it's bound. And the question is, if we can follow this change, how did it go from here to here? Okay? Can we follow it as a function of time and see how it evolves the minute it binds an oxygen? Okay? So, what we want to know, what people are interested in, is figuring out how the structural changes that take about when you go from the T to the R or from the R to the T, when you change from these two structures. So, this turned out to be very difficult. Okay? And one of the reasons is, so people have done a lot of work trying to answer this problem. And it's difficult because the structures that you're looking at are structures that are very short-lived. They're non-equilibrium structures. And they're very short-lived, meaning they could live maybe 100 nanoseconds. Okay? Microseconds. Okay? So it's very difficult to follow them. And in solution, we have a difficulty that we call the diffusional barrier. And that is that by the time so let's say one of you guys come to my lab. Okay, actually I have 3 students now working in the lab. I don't know if they're here today. Okay, 3 students from this overall group. And let's say one of you guys are working in my lab and you want to study this. Okay, so you say, okay, so I'll take the hemoglobin, I'll take some oxygen, or in other words, you just expose it to air, because air has oxygen, and I'll see the oxygen will bind, and I'll follow the structure using this microscope. Okay? So how quickly can you mix the oxygen into the solution? Okay? Let's say you're super quick, and you just open it and quickly mix it, and then quickly put it in the instrument to measure. How long would that take? Seconds? Okay? But it takes for the process to change, it takes maybe 100 nanoseconds. Okay? So in that 1 second that you take it, the whole process is over with. So how can you study it? Right? You see the difficulty? And it's not just for this, it's everything that involves mixing something into solution, whether we have to change it, the structural changes you want to follow is very rapid. Hundreds of nanoseconds. So a nanosecond is 10 to the minus 9 of a second. Okay, so imagine 10 to the power of 9 seconds pass until you actually, by the time you finish mixing it. Okay, there are other methods, there's rapid mixing method, and there's different approaches, but they still have that limitation that is dependent on how long it takes for them to mix it. So one approach that we have developed in the lab at Einstein and also now used in my lab is looking at using a method called soul gel. Okay? Fabricating these matrices. So in this method, you take our starting material, which is, for example, tetramethyl orthosilicate. So it has this molecule, it has a silica, and then these side groups on top of it, and you mix it with either acid or base, and in that process, a reaction takes place, a polymerization reaction takes place. We have first have a step of hydrolysis, then condensation, and eventually polycondensation, and you form a gel. Okay, so you form a solid matrix, okay, and that matrix has water in it. It's very porous, it's transparent, it's not toxic, and it has other benefits that I'll mention later. So this is known for a while. You could make that, and people fabricate a bunch of different things. Okay? At the end, what you have is a network of silica, oxygen, similar to sand. Okay? So it's a silica matrix. And that's why I say playing in a sandbox. Okay? So eventually you start with these precursor and you end up with a network of silica, oxygen. So what will happen if in that process when you take the solution and let it gel form a solid, okay, if you also add a protein. Okay? So if you add a protein in this process, okay, it will get embedded into that solid. So you have a network of the silica oxygen and in it some protein. So you've encapsulated the protein inside that solution. Okay? So what are the advantages of this kind of an approach? Okay? So the sol gel is transparent, it's inert. Okay? It doesn't affect the protein. Okay? And we can, in most cases, the protein that's intact, they're not damaged by this process. Okay? They're still functional. For example, hemoglobin can bind and release oxygen. And they actually are rendered, in some cases, more stable than the solution. So you can take that and you can heat it up to a high temperature and nothing happens to the protein. Okay? And the other advantage is the protein in this condition, it's stuck wherever you trap it. It cannot diffuse, it cannot move. Okay? In solution, the proteins, they just move around. Okay? In the gel, they're stuck where they are. Okay? So one of the problems that you can have with study protein is that you put them in solution and you're going to say, okay, let's see what happens if I go to a very low pH. Let's see what happens to the protein. I want to study the structure of a protein when the pH is very low. So you change the pH to a very low, and the proteins start clumping together, forming a big aggregate, and they go out of solution. So you can't study them. So in the gel, they can't aggregate because they're stuck inside of a certain location. So then you can study them under these conditions. So these are some of the advantages, and maybe the biggest advantage is that the sol gel is very porous. It has tunnels that run through it. You can think about it as if you have a sponge that you are used in the house to clean the surface of by the sink. Okay, that you can take it and soak it in water and then squeeze it and the water comes out. So this is something similar to the sol gel. It sucks up water. It's very porous, so it gets filled with water. And the protein that's stuck inside the sol gel, the water molecules can diffuse in and surround it. So that the protein feels as if it's in solution, because it's surrounded by water within that transfer. So what we could do, for example, we could fabricate to make a film of that sol gel, have it on the side of the cuvette. Okay? For example, over here, this is the cuvette, this is the gel, this is what it looks like. Have you seen the cuvettes before in the research? Okay, so this is just a standard cuvette. You cast the film of the sol gel over here, and in this case, we incorporated myoglobin. Okay, so now the myoglobin is inside here. Okay, that will give it the brownish color. This is metmyoglobin, that's one of the forms of myoglobin. Okay, and then we could do a bunch of different spectroscopies on this gel and essentially measure the properties of the protein. Okay. And this is an example of the spectra that we took, UV spectra of the protein when it's in solution, which is the top, or when you took it inside the sol gel. And you can see that it looks the same, which tells us that the protein has not done any changes. It still is, the structure is intact. Which is important, because if we put it in the gel and the structure changes and we didn't gain anything, right? We want to study the structure of the native protein. So one of the advantages of this method is that it allows us to overcome this diffusional barrier. Okay? And the reason for that is that the sol gel environment is very viscous, okay? And it slows down all changes, that structural changes that could take place, any conformational change. Okay? And it slows down the diffusion of ligands in and out. But it also slows down, and more so, the changes of the protein. Because the protein is much larger, and